Thank you for having me, especially under these strange circumstances that at the end when this meeting started, I was not around. However, when I heard about this meeting, it was very important to me to share my own experience from the field of cryo-EM on the issues which related to reproducibility of results, and actually even one step further, how do you know that the experimental system that you are using, or what assurances do you have when the experimental system that you are using provide meaningful data? And in cryo-EM, this question is very, very crucial, since I would show the pitfalls of the technique in a second and how we arrived to our molecular structures, but I will give a short introduction actually to what is cryo-EM. So today, cryo-EM is the highest resolution imaging modality that humanity has. Everything that any molecular structure that we would like to really see, to watch with our eyes and image, the only tool that we have today available to us is for getting to atomic resolution while imaging is cryo-EM. There are other methods that will be spectroscopic methods where some of the information is inferred via other methods. However, cryo-EM is a microscopy method. In other words, we are striving to get, and we are getting closer and closer to the day that we will be able to slice a piece of a cell, look inside it, and see the molecular structure on atomic details inside the cell. So on the left, you see one of the world records in resolution, where you can place inside the output, the final output of this method would be these electron density map described here as the blue, green, and yellowish webs. And when the resolution is high enough, in principle, a person can go and track these clouds and start placing atom by atom, and therefore get a very clear understanding of what is the chemical model that underlies biological phenomena. And why is it that important? So immediately after breakthroughs that happened around, that culminated in the 2007 Nobel Prize in chemistry, it was clear that the path for understanding the interaction of molecules one with each other, in this case an antibody and the Omicron spike, and the path to understanding the interaction between drugs and proteins, which is extremely crucial to our ability to develop new drugs, is now open. Up until to that point, the only way for us to really be able to do that was by crystallography, another very high resolution technique. However, this technique requires dramatically more extensive work while preparing samples and put them under conditions which many times would be disputed whether the final structure that we get is actually representative of the reality. While what I would show here, that cryo-EM in principle can bring structures that we are very sure of their, if we get the structure, we are very sure of its correctness or the representation of reality. However, sometimes we have questions whether the structure itself is correct. In other words, the chemistry and the biochemistry are kept. However, the way that we deduce about it is sometimes have to be scrutinized. And I'll try to explain why. So as I said, cryo-EM was a niche, a tiny technique that was among the biochemists called blobology, because what normally we would get for years from the 1950s up to 2014, this type of very low resolution blobs, and we would need other techniques like X-ray crystallography or NMR in order to dock into these blobs high resolution information. This stopped being a niche technique due to several advances, all of them due to this nice person here at the bottom, so Richard Anderson, Jacques de Bouchet, and Joachim Frank. and which awarded them the Nobel Prize for their huge contribution over many, many years, and actually a lot of resolve and resilience. And technically, the changes came with new types of detector allowing to get much higher and better information from the sample, and the immense progress of computational tools, as you would see very soon. In order to achieve a structure, a three-dimensional structure, this is extremely computationally heavy field. So now, the main thing that we'll be interested in when we are looking in any microscopy modality is what is the resolution. Now, the classical way to look at the resolution, it comes from the understanding already from Abe many years ago, is that any microscopy modality would reduce the amount of information, is actually cannot recapitulate all the information that is coming from the experimental system. And that results in the fact that no matter how much you make things larger on our screens, because information is lost, we would get a blurring of the data, blurring of the image. And when the image is too blurred, if this red point and green point were the same color and were very nearby, you will never be able to separate them. And as a result, this blurring sets up the instrumentation resolution limit. However, there is a clear separation between instrumentation resolution limit. I can actually today with electron microscopy build electron microscopes that can reach to a resolution that is subatomic. However, all the electronics, all the physics are set. We know how to do that. However, the sample is another source of losing resolution. And that is due to limitation that the sample would not allow us to collect the data to the highest capability. And that's a direct result that we would destroy it while we are collecting the data. And as a result, even if we have high resolution terms in our data, when we come to average many, many noisy images, the results are poor. And as a result, one of the most important things in our field is knowing what is the resolution that is for the current experiment. And I can show here, I'm showing here 4 different examples of what are results at different resolutions. So from the 20 angstrom roughly globology situation that I showed before, up to 2.6 angstroms. And actually the first figure in my talk was at 1.7, which I think is now the world record for cryo-EM. And you can very easily see that the information content decreased dramatically as we move with the resolution. And why is that important for us? Because actually the resolution criteria that we would use determines the boundaries of overinterpretation. And this is directly related to the issues of the field. In principle, first, when people publish papers, they will never write, here is the low resolution structure of the coronavirus, since people would be less excited to read these papers. But other than that, something which have a practical meaning, if I put here a drug, for example, where my cursor here, and I would like to understand the interaction between the drug or the protein, or if I would like to claim that there is a water molecule here, if I claim that the resolution is very, very high, and it's not, then a blob of something that I see here is actually a piece of noise, and which means that I'm over-interpreting the data. So the common practices in the field is to try to assess the resolution and then filter down the data to delete the high resolution terms from the data. And that is done with set of preset and very known image filtering tools. However, if I claim my resolution, I would filter to a higher resolution, which means that pieces of noise will stay in the data, and then I can commonly claim that I can see, let's say that here I see some kind of an ion that is bound to the protein, and that is actually very important because if you think about it, these structures are from one side the strongest drivers for interpretation of biochemical phenomena and drivers for the different assumptions about biochemical systems and how do they behave. On the other side, from the point of view of industry, these are crucial in order to understand what is the next step on development of a new drug. I hope at that point, I would like to know, because it's very hard for me to see is whether it is clear that the problem is stated clearly. In other words, we are extremely interested in what is the resolution of an experiment. It is today completely, for our field, not determined by the behavior of the machinery. The machinery actually, if you would have a very good and resilient sample, would get a resolution which is dramatically beyond what we would need in order to interpret biological structures and the problems which are associated with them. However, due to sample limitation, our ability to extract the highest quality information from the very, very high quality machine is many times limited, and we need to interpret and decide what is the resolution that we have in our sample. So that is one problem that would loom all over what I'm saying now, and I'll discuss towards the end of the lecture what are we doing in order to do that. And now I'll go to some further details of how do we do our work. Because you would see that would explain why the question of resolution is there, and actually even bigger question, the question of correctness. If I'm saying resolution is being sure that what I'm seeing is correct at a given level of details, there is another question. It could be that all what I'm showing is wrong. So that's the difference between knowing that what you are looking at is a book and not a box. That's correctness. While resolution is being able to see already what is written in the book and interpret what you see. So this is knowing that you look at the book and then understanding the high details that you see in the book. So in principle, the two most used methods in order to gain structures in cryo-EM are shown here. The top one is called single particle analysis. And in single particle analysis, we take many pictures, what you see here, of molecules which are frozen in time and in hydrated ice. Since proteins, their interaction with water is crucial to their structure, we just stop water from moving at very low temperature by doing very quick freezing, and that goes under these cryogenic conditions into the microscope. And the assumption when we do this data collection is that all these molecules are actually the same. And if these molecules are not the same, when we will try to get a structure, each of these molecules look very nice to the human eye here because they're filtered. Normally, they are very noisy, and it's very hard to see them. And we pick them one by one today, most of the time using experimental, like automatic machines that would select them. And for each project, about 1 million particles would be picked up. So it's a lot of computation that goes in. And these are averaged, and after averaging, the noise is averaged out, the data is averaged in, and at the end we have a bunch of nice two-dimensional projections. But we need to go from the two-dimensional projection to a three-dimensional structure, which means that the computer will have to solve with human assistance this three-dimensional puzzle. And this is a very tough computational problem, and I'll give an example to explain why is it a tough computational problem in a second. Another way to do that is with tomography. And in the case when you do tomography, you collect, similar to a CT scan, but in this case, the patient is moving and the machine is static. You collect many images of the same object from many directions. And box out, you produce a three-dimensional structure of that. In this case, this is a part of you see here an HIV viron, and you see the spikes of the HIV on it. You box out these particles, align them, average them, and you get a nice structure that you are looking for. In both cases, the data is extremely noisy. At the bottom case, the data is dramatically more noisy, and everything is limited by the ability to expose the sample. Since the radiation that comes from the microscope is, for any mean from biological perspective, is identical to any other ionizing radiation, like radioactive radiation, it actually breaks down the sample under the microscope beam, and as a result, we are actually picturing everything here with very low number of electrons, which means that in order to solve the three-dimensional puzzle or to deduce the three-dimensional structure, it's very similar that I would take a picture of a room almost with no photons. I can barely see the people in the room, and now I'll have to decide which person is which, or which chair has a person in it and which chair is empty. And so clearly, as the amount of photons that I'm collecting getting lower and lower, this job becomes harder and harder. So how do we solve this issue? In principle, it's already clear that if I would like to solve this three-dimensional puzzle, if I had very clear pictures of the room, for example, it will be much easier to align the pictures to each other than if I have very dark pictures. But I'm pushed to have dark pictures since we cannot collect with high doses. That would destroy the structure. So there is some kind of chicken and egg problem. Had we had a structure, it would be very easy or easier to align pictures to the structure. However, since we don't have the structure, this is our scientific question, we would have to take very noisy images and try to align them without knowing what we align. So it's like solving a puzzle in the darkness. When the puzzle is getting solved, it's becoming easier and easier to continue solving it. But the beginning is very, very hard. And this is a typical type of an inverse problem. So from non-mathematical perspective, but you can clearly see how statistical tools can be drawn to this type of a question. If you wake up one day and the door at your home is really like going to fall off because somebody is banging like a crazy on the door, and then you have to decide what would you expect to find on the other side. So this is a classical inverse problem, which is to reconstruct a high-dimensional causative agent. In other words, what was the thing on the other side of the door? From very low-dimensional observation, you only hear the sounds of the banging and you see the door jumping. Now, inherently, one of the problem is that if the door is being banged like that, it could be many, many explanations. So for one example is that the Girl Scout at your neighborhood are banging at your door and they were very eager to sell you some cookies. A more probable explanation would be that it's your neighbor. So it could be either very aggressive Girl Scouts because the door is really jumping, or it could be that you double parked and your angry neighbor is actually want you to move your car. So what I show with this example is actually that we always, in cases that we solve an inverse problem, we actually go the opposite direction of normal experimental tools, and it is in many senses it encapsulates actually the weight of all scientific activity. In other words, we see signals in reality or in our experiment, and with that we try to deduce a model of what generated these signals. So what I like many times when I think about the philosophy or also the mathematical foundations of cryo-EM, that it is encapsulation of all scientific activity in one type of experiment. In other words, we bring up signals from an experiment. They are always just a single representation, single manifestation of reality. And from these signals, we try to deduce what is the underlying story, in our case, a three-dimensional structure that, or a set of three-dimensional structure that can explain the results. So prior knowledge is a must, because if you know that you have no monkeys in your neighborhood, but you have a lot of aggressive scout girls, so that should be the answer, okay? So the prior is a crucial thing for us to be successful in solving an inverse problem. However, if you don't know what is the structure, then from where do you bring this prior? And this is the biggest issue in the field, and you would see very soon why it's called the Einstein from noise problem. On top of that, you can already understand that if I would like to place many different projection images and decide what are the directionality solve this structure, this is actually an unusual fit problem. And the reason that I say unusual fit is because what you see here is a classical experimental statistical fitting problem. We have some kind of an underlying model. It doesn't really matter what are the mathematical details of the model, and my student goes to the lab and do an experiment and come up with all these red points and then say, okay, let's fit the model. So the student would not like these outliers. And then you can ask yourself whether the model that in this case come from a Hill equation from many, many years ago is the correct model, or so there are several things you can do. You can repeat the experiments. Hopefully you'll get more points around here, and you would either have to declare that these are outliers and explain why you got them from the first place. You can be a really bad scientist and just delete these points, or you can be another type of what will be considered a bad scientist, and you will do overfitting. You'll say, okay, now I have a new curve, and let's put this curve and explain why this model is correct. Now, the good thing about this experimental system that if you are not happy with your results, you can go and do more and more and more experiments, and hopefully you will either see that more and more points fall in these unexpected areas, which means that you find a new phenomena, or after many, many experiments, you would get enough data that will be certain that the fit to the orange line is the right fit. However, this type of experiment is easy because the number of fitting parameters is always kept small, and the amount of data can get larger and larger and larger as you wish. It could be that as you wish is very expensive, either with time or with amount of other resources that you'll have to put. But in principle, this is a solvable problem, which means that at the end you will be able to decide whether the green one is a bad fit or the orange one was a bad fit, and maybe you need another problem. Now, it could be that the student is very clever, very resourceful, but not very, but quite lazy. And he would come with a different type of experimental machine. He said, look, I'm fed up with measuring all these points all the time because each one of them takes me many, many hours at the lab. I invented a new experimental machinery. I can measure many points, noisy points on the same curve, but there is one issue. There is a problem of calibration. Somehow these curves somehow fit to the same story. So how do we go around that? In principle, it's enough that we'll bring the student a single prior, and we'll be able to put all the curves together, average them, and then get a very nice signal. But let's say that the prior was that most of the curve must pass through this area, and if the prior were wrong and we would put the curves here, we would get everything shifted, which means that we'll get a really, really wrong answer. So I show here that there is another way to go around an experiment, build up an experimental system that measures many noisy points and find a prior that will help you to average the data. So this is exactly what we are doing. In principle, we will need to find the position parameters in shifting and space, and in angles of every measurement that we done. So in principle, we would like to know where the image is shifted, how it is rotated. In 3 dimensions will be 3 angles, not 1 angle, and many other things that come from the experimental system that I would not go into, but you can see that the list of fitting parameters is getting larger and larger as you want to get to better structures. And there is another case that it could be that one of the proteins actually not the proteins that we are looking at, but a similar protein or a different conformation. So this one is not smiling, and we need to be able to separate it out. So it's a very hard fitting problem with many, many fitting parameters, about 1 million or so for every experiment. On normal experimental methodology, this is completely unacceptable. And like fitting would be done with 3 parameters, 5 parameters, much larger than that. You can fit, in principle, everything. So another problem with this experimental method that unlike the original experiment that we devised, each time you will add an image, you'd say, okay, I'll do another experiment. I'll bring another realization of the system. I would get another set of fitting parameters, which means that the number of fitting parameters in this case grows as we do more and more experiments, which is completely against what I showed at the beginning with the orange curve. Now, that could lead to ridiculous results, which is known the Einstein from noise problem, and the field is very much aware of it. You see here Richard Henderson as the first person written here, and Joachim Frank here. And the reason is that I can't completely, if I take a very strong prior and I take a data set that is completely has no data inside it, and put a prior of Einstein, I'll get Einstein from the data, and it's quite clear that we didn't have Einstein on that day inside the microscope. So how do we go around it? We have several tools that we are using in order to check that we are not overfitting. In other words, assuming new information at high resolutions on one side, and we are giving the correct structure. So the way that the field goes around it is mostly frequency matching. In other words, delete high frequency information in our data, solve the structure, and then after you solve the fit, you have the fitting puppet, add the high resolution terms in order to see whether they're making sense. And in order to check whether they're making sense, we go back actually to the orange curve. Instead of doing the experiment once, we split the data to 2 halves and solve it once here, another one, and then we compare them in frequency space. And these are the curves that actually would be used in order to show now every researcher. It became a standard that every researcher that does the experiment have to show whether these curves of correlation between the 2 halves of experiments make sense. Now, what I'm pushing in my lab is to add additional external information on the structure after you get to high resolution. Since we know that there are chemical fingerprints to our data, in principle, we can carry, and the chemical fingerprint that I selected for doing that, actually that's the reason that I'm in the US, is to propose that as a standard method, is to see that because we know that different chemical elements of protein structure decay differently under the electron beam, we can also put the decay as an information. And what we are proposing is actually to use the decay information post gaining the structure. In other words, the statistical tools that would lead to the three-dimensional structure will be oblivious to the chemical information that they're looking at, which means that we have an orthogonal piece of information to validate that the glutamate in a place is really glutamate because it decays quite quickly, while histidine, which decays slowly, is histidine because you see that it is decaying slower. And the colors here represent the rate of decay from red to blue. And is it adequate? For most part, yes. We do see some cases where it is very hard to validate the quality of the results, but on non-pathological cases, if you keep the books correctly, you can avoid the Einstein from noise problem. And thank you for your attention. And I hope that I was clear enough of what are the issues and how do we solve them.
Thank you, Gabi. We have one question. I mean, we have probably more, but time for just one short question, so the others don't have to. Or shall we just go on? And okay.
No.
Yeah. There's one question.
Is the cooling condition or the cryo, isn't it also a limitation because you may freeze structures, movements which actually exist in the real stuff?
So the sample preparation problem is even a little bit wider than the cooling problem. There is a danger in the field that when you freeze something, and since it's the closest to native conditions that we could ever get outside of a cell, there is this easy assumption that what you did not affect the structure. And that's not always true. And we know, for examples, that pieces of the proteins would disappear, things would fall apart. It's actually not due to the cooling itself, because it's extremely quick, much faster than most of the reactions that happen, but the condition just before the cooling. As for the cooling, there is a whole research that is going on what temperature is the system is representing after cooling. And there are another set of very complex statistical tools that is now being set in order to be able to solve not a single structure, but a continuum of movements, and that would directly answer the question that you asked. And the jury are still out. Like, this is one of the central discussions that are done today. How do you build a mathematical representation that will take this sparse data that is rich with details but is all covered with noise in order to bring up actually the details of what you're saying? What happened to the molecular vibration when you stopped all movement? I hope that was clear.
Thank you very much. Our next